Welcome, everyone. My name is Tayfun Tatar. I'm an application scientist at Hansa Biomedical Life Sciences, one of the pioneers in the field of extracellular vesicles. Before I start my presentation, I would like to thank Dr. Radwa Mehanna and Dr. Eman Thabet on behalf of all the members of Egyptian Society of Extracellular Vesicles for inviting me to this presentation, even though it is online. I would also love to be there, considering that it is around minus 15 degrees right now in Tallinn, Estonia. But here am I. So today I would like to talk about a next chapter in EV isolation through tangential flow filtration, one of the novel approaches. And during my presentation, I will first start with an introduction into extracellular vesicles, but I will keep this short because I'm aware that I'm already speaking to an EV-focused audience, so you know much better than me. But I will continue with the isolation aspect of EV research, and I will start with why and why not ultracentrifuge, speaking of its advantages and disadvantages. And then I will slightly move into tangential flow filtration. I will first give a technical background of it. Then I will focus more into its chronology, how it got implemented into EV research. And then I will also talk more about the technical aspects, namely the advantages, features, and some literature examples, both from review point of view and also from case study point of view. So let's start with an introduction into extracellular vesicles. Extracellular vesicles are cells' way of communicating with each other through the functional molecules that they carry within their cargo, which includes proteins, RNAs, or DNAs. And they are highly heterogeneous. They can be classified based on several different criteria, and these include size, cellular origin, cargo, or cellular function. And they are available from many different sources, which can be biofluids, cell-conditioned media, plant extracts, bacteria, fungi, or algae. And together with the heterogeneity, this gives rise to the question how to obtain or purify EVs from these sources. This plot here shows the number of publications using different isolation methods for isolating EVs from their sources. Just as a note, these do not include all the available EV isolation methods, but this is just a representation. But you can still see that ultracentrifugation is by far dominant. So why ultracentrifuge or ultracentrifugation-based EV isolation is dominant in the field of EVs? Because first of all, it was not invented solely for EVs, but it was a widely known technique for years. Therefore, it was much easier to come up with an established procedure for EV isolation based on ultracentrifugation. And also, it doesn't involve any chemical modification, meaning that you don't affect the physical chemical characteristics or the functionality of EVs, which is critical either if you are working on identifying the functionality of EVs or you want to use these for downstream applications. Therefore, over time, ultracentrifugation became the gold standard in EV isolation. But this does not necessarily mean that EV isolation through ultracentrifugation comes without any risk or any disadvantage. The first disadvantage is damaging the EV integrity because ultracentrifugation applies a high shear force on EV membranes, leaving these membranes fractionated or damaging EV integrity in short, or they can even cause aggregation. And these two disadvantages are critical, especially for the functionality of EVs, because if your EVs are not intact, they might not deliver the same functionality or with the same effectivity. And then there are also disadvantages from user experience point of view, including its time-consuming nature, meaning that it requires hours to process even minor amounts of sample, and it is also labor-intensive, especially for sample preparation perspective. And finally, it is not a scalable method at all, which is a barrier, especially for end applications, whether clinical or industrial applications. If you need to process more volumes, the only thing you can do is to purchase or acquire another ultracentrifugation equipment. And as a result of all these advantages, now ultracentrifugation is actually questioned a lot, even though it is still maybe the gold standard in EV isolation. It is not an unquestioned method anymore. And this is just an example from January 2026 in a review paper that discusses EV isolation methods for transition from basic research to industrial scale. This review paper did not even include ultracentrifugation because of its well-recognized limitations. So in short, in two different aspects of EV field, the basic research requires cheap and fast solutions with high purity, whereas end applications require reproducible and scalable techniques that can provide high yield. And this is where we will start introducing tangential flow filtration. So first of all, the technical background of tangential flow filtration, which is also known as cross-flow filtration. It is a technique based on filtrating the sample in the perpendicular direction to a laminar flow, as you can see also in the schematic illustration on the right, whereas the flow direction is from left to right, filtration direction is from top to bottom. And this method is a widely used method for years in biopharmaceutical or food industries for purification, concentration, and diafiltration of biomolecules within biosamples. To give a bit more details, this method is based on sub-millimeter diameter fibers, as you can see again in this schematic view. Sub-millimeter diameter fibers made of non-binding materials such as polyether sulfone, polysulfone, or regenerated cellulose. And all these fibers have certain pore size which determine the cutoff of filtration. Let's say if the pore size is 50 nanometer, particles smaller than 50 nanometer are filtrated out, whereas those larger retain inside. So it is a size-based filtration method. And here I hope you will be able to see this animation. In this animation, you can see how it can be used manually in practice. You basically connect your syringes on both ends and you push the sample back and forth until there is no filtrate coming out. Just for visuality, I can show once more. So this is how simple it can be used in practice. So in a sense, it is an ultrafiltration method, but what makes it different or more advantageous than dead-end filtration? In dead-end filtration, there is the well-known phenomena of cake formation, which is basically particles larger than the pore size of the filter accumulating on top of the pores over time. And then as this cake thickness grows, the flow rate through the filter decreases, meaning that the efficiency of filtration decreases. But in the tangential flow filtration, since the laminar flow is perpendicular to the filtration direction, this ensures a steady filtration over time, maximizing the efficiency of filtration or EV isolation in our case. But of course, as I also mentioned earlier, TFF as well was not invented for the field of extracellular vesicles. On the contrary, it has been also a widely known method for the life science researchers. During 70s or 80s, it was developed as a technique to purify proteins or separate cells in research settings. But then over time, as it became a more of an established method, it was also introduced in other fields, including food sciences and biopharmaceuticals. And it was only around late 2010s that it was also started to be embraced by EV researchers as an alternative method to isolate EVs. But after this introduction to the EV research settings, we can claim that it was embraced quite rapidly, because if you consider the plot on the left, which shows the total number of publications performed through EVs isolated with TFF, we can see an exponential trend in this total number. And we can even expect a sharper increase from this point on, because in the beginning of 24, 2024, updated MySav guidelines included TFF as an alternative method to concentrate EVs, meaning that now TFF is officially recognized by EV researchers. So up until this point, we already covered what EVs are, why it is challenging to isolate EVs, or why do we need alternative methods. And then we introduced EVs. So now it is time to answer why TFF can replace this unmet need in the field of EVs. So for this purpose, I would like to first start with the advantages of TFF. So the four most pronounced advantages maybe of TFF are, first of all, reproducibility. Because this reproducibility is something that minimizes your batch-to-batch variability, which is critical both for research settings if you don't want your duplicate experiments, for instance, to vary in terms of the results just because of the isolation method, or similarly for industrial applications, if you want to make sure your EV-based product has similar quality as the previous batch. So this is a key in standardization of your results. Then a second critical advantage is easy scale up, because contrary to ultracentrifugation, for instance, if you want to scale up your EV manufacturing, the only thing you need to do is to change from a smaller module of TFF cartridge to a large module, because while doing so, you don't deteriorate or you don't compensate from your performance. The fibers are the same, the filtration principle is the same. You can only process larger volumes. So this is a very easy approach of scale up. Then there is also the reusability of TFF cartridges, which allows it to be reused multiple times in between each use just by washing according to the protocol by the manufacturer. But here it is also critical to worth mention that this reusability is not coming with a certain number of reuse, because it depends on multiple factors. These include some biological factors such as sample type, sample contamination, or even storage conditions, pre-clearing steps. If it is cell-conditioned media, for instance, the conditioning material. So because of these multiple factors, some of which are out of control, it is impossible to define a certain number of reuse, but they are reusable multiple times. And they are even sterile, allowing to be used under the hood. And even after the sterility is broken, they can be re-sterilized through e-beam or ethanol oxide sterilization. And by the help of all these advantages, if you compare it with the gold standard, which is maybe the first step you should do if you are introducing a new method in any field, the comparison drops down to this table, more or less. So first of all, ultracentrifugation results in sticky EVs with fractionated membrane, whereas tangential flow filtration ensures smooth flow of particles across hollow fibers with minimized aggregation. In ultracentrifugation, the recovery of EVs is very poor, limited to around 30 to 40% in most cases. Whereas in tangential flow filtration, you can ensure even more than 80% of recovery. Ultracentrifugation is a time-consuming method. Even to process a couple of tens of milliliters or hundreds of milliliters, you need hours, whereas in tangential flow filtration, you can do so within minutes or on the range of tens of minutes. Ultracentrifugation does not allow scale up. The only thing you can do if you need to process more volumes is to buy a new equipment, which is not very cost effective. Whereas in tangential flow filtration, you can process from a couple of milliliters to tens of liters just by changing your cartridge. In ultracentrifugation, the contamination is a bigger problem because there is no sharp cutoff introduced from density perspective, from size perspective, or from any other perspective that can be determining EVs. Whereas in tangential flow filtration, there's a sharp cutoff introduced by the pore size of the fibers, minimizing the contamination. For instance, if the pore size is 50 nanometer, you can minimize the contamination coming from particles below 50 nanometer by this sharp cutoff. And finally, ultracentrifugation requires highly expensive lab equipment, which minimizes the accessibility, especially for newcomers to the field. If you want to develop a new lab, this is a very big consideration. Whereas for tangential flow filtration, you can use these filters even manually, or if you need automation, you can do so by a simple peristaltic pump that is widely accessible. But of course, the comparison is not only when you compare it with the gold standard, because the researchers have multiple isolation methods at their disposal. So you need to also provide with some benefits also compared to other methods. So the standalone advantages, or one of the standalone advantages of TFF is its versatility, because the versatility allows it to be used in many different scenarios. You can use the very same TFF cartridge for separation or isolation of small or large EVs, concentration of pre-isolated EV samples, buffer exchange, removal of contaminants, EV depletion, or elimination of unwanted proteins. And here you can see schematic illustration of some such scenarios. On top left, for instance, you can see that two TFF cartridges with different pore size, namely one with 200 nanometer and one with 50 nanometer, can be coupled in series to isolate outer membrane vesicles from bacteria cultures, sizing between 50 to 200 nanometer pore size. And you can apply this similar schematic to biofluids, plants, fungi, or whatever EV source you might want to target. On top right, you can see that it can also be used to deplete FPS to remove unwanted components, after which you can use this depleted FPS in your cell culture work. And this is a critical advantage, especially if you consider the much higher cost of depleted FPS compared to undepleted one in the market. So you can just purchase undepleted FPS and deplete FPS at home by the help of TFF. And then in the bottom, you can see that TFF with, for instance, 5 nanometer pore size can be also used to concentrate pre-isolated EVs, which is critical especially for in vivo or in vitro experiments where the control of dosage is critical. But this doesn't mean that you can concentrate only one type with only one type of TFF cartridge, because this is also another advantage of the technique, that for instance, even 50 nanometer or 200 nanometer pore size cartridges, they are concentrating the sample while simultaneously isolating EVs. So you can actually reach much higher efficiency in terms of your workflow this way. But the versatility is even not limited to the standalone use of TFF, because it can even be coupled to other methods by the help of its gentle approach. And this coupling can be done either with other size-based approaches like size exclusion chromatography or ion exchange chromatography, for instance, as an orthogonal method. And regardless of the method that is coupled with TFF, the advantages that TFF brings into these combinations is, first of all, the concentration of EVs, meaning that while removing both small solutes and large debris, TFF can concentrate EVs from much larger volumes, even tens of liters, depending on the scale of the TFF cartridge, to very small volumes like a couple of tens or hundreds of milliliters, with which the subsequent method can work. So normally, size exclusion chromatography columns cannot work with or cannot process tens of liters, but after processing and concentrating with TFF, you can also employ a size exclusion chromatography for further purification, and this ensures the scalability and efficiency of your workflows, even if they require high purity. And even further, apart from the technical aspects of it, let's say regarding the versatility, regarding the purity, regarding the yield, up until now, I mentioned always that it can be used manually, but especially if you want to process large volumes, not a couple of hundreds of milliliters, but liters or even tens of liters, of course, it might not be so suitable to just connect two syringes on both ends, but you might require some automation. And currently at Hansa Biomed Life Sciences, we are building our own automation system, which is compatible with all our TFF cartridges. And the aim here is to allow EV isolation in a much more controlled way through rapid workflows. And this will minimize human intervention, increasing the reproducibility of results even further. So after covering all these advantages, now I would like to also focus on some real examples. And these real examples will start with some review examples. So I will try to give a grasp of how the researchers position tangential flow filtration in EV field. And then I will also talk more about some case scenario examples. So as for these review examples, I have three examples here. So on the left, you see a review focusing on plant EVs as the next generation therapeutic agents or drug delivery carriers, which mention that their advantages over mammalian counterparts include high biocompatibility, productivity, and effective cellular delivery. However, this review underlines that in order to exploit such advantages, we need scalable EV isolation methods such as tangential flow filtration. Then the review paper in the middle focuses on EVs in a source agnostic manner for their promise in drug delivery. But this review mentions that either for preclinical applications or studies or for clinical trials, one of the biggest challenges is producing EVs in large scale because of the low yield and contamination problem of conventional methods. And this paper also nominates TFF to improve the quality of isolated exosomes or extracellular vesicles. And then on the right, we see a review paper focusing on another potential application area of EVs, namely regenerative medicine, which can be reached through like stem cell EVs, plant EVs, or any other source of EVs. But here, the problem is that despite the satisfactory purity that can be reached through ultracentrifugation, the low production yield and long processing time makes it difficult to reach the desired production configuration for such applications. And this paper as well points to tangential flow filtration as a widely recognized method to overcome the present challenges in EV isolation. So as you can see, overall, the main advantage or main positioning of TFF is to enable transitioning from basic research to application stage, whether clinical or industrial. And finally, now let's move on to some case scenario examples, meaning how researchers use TFF in their studies. For this purpose, I will present you with 5 different examples, each focusing on different aspects using different source EVs. The first example focuses on induced pluripotent stem cell-derived mesenchymal stem cell in comparison with primary human umbilical cord stem cell derived EVs, claiming that induced pluripotent stem cells have higher immunomodulatory and regenerative potential. And in order to prove their claim, they isolate their EVs. Then they use TFF with 5 nanometer pore size, named TFF Easy in Hansa Biomed catalog, to concentrate these EVs before in vitro injection, for which the dosage is critical. And through in vitro scratch assays, the researchers were able to demonstrate that induced pluripotent stem cell EVs were indeed showing higher pro-regenerative potential compared to their human umbilical cord derived counterparts. Then in the second example, the starting material is now bacterial cultures, with the idea that outer membrane vesicles from bacterial cultures are promising platforms for vaccine development or drug delivery, but the challenge is measuring their biodistribution. Therefore, these researchers aim to come up with a highly efficient radiolabeling strategy for in vivo imaging studies. And for this purpose, similarly, they employ TFF with 5 nanometer pore size for concentration of EVs prior to injection into in vivo mice models. And through this in vivo studies, they were able to optimize their protocol based on high nitromycin to enhance radiolabeling efficiency during biodistribution measurement. In the third example, now the starting material is a plant extract, namely citrus, with their EVs offering an excellent platform for nutraceutical delivery with endemic cargo full of micronutrients. However, the problem or the challenge is their inherently unstable cargo, which is prone to storage or preservation conditions. And then here, the aim of the researchers is to develop a standardized storage and preservation conditions for which they use two tangential flow filtration cartridges in series, namely one with 200 nanometer pore size, namely TFF-MV in Hansa Biomed catalog, and one with TFF, one with 5 nanometer pore size, namely TFF-Easy in Hansa Biomed catalog. So they use TFF to first isolate EVs from citrus extract and then to concentrate it before in vitro assays. And then they perform in vitro assays under different storage and preservation conditions such as fresh juice, frozen juice, freeze-dried juice, and so on. And through these in vitro assays, they were able to optimize their protocol for standardized storage and preservation to maximize the physicochemical and antioxidant activity benefits from citrus EVs. In the first example, now we see the combination of TFF with another method, namely size exclusion chromatography, for studying the role of small EVs in human limbal niche homeostasis. For this purpose, researchers isolate EVs from epithelial, stromal, and melanocyte cell cultures, first by concentrating the conditioned media through TFF with 5 nanometer pore size, namely TFF-Easy, and then by isolating or purifying further with size exclusion chromatography columns. Since this combination is a gentle approach, on the contrary to ultracentrifugation, this approach allows downstream analysis, namely proteomics analysis, which suggests the researchers, that small EVs may be regulating keratinocyte development, extracellular matrix organization, and regulation in human limbal niche homeostasis. And then in my fifth and last example, the starting source is now bovine milk, which is an abundant, cost-effective, and eco-friendly source of EVs with demonstrated potential in chemotherapeutics or bioactive natural compound delivery. However, the challenge is again the scalable production of bovine milk EVs with an optimized protocol. And here in this paper, the optimized protocol or the proposed protocol is enzymatic casein removal, which is an important step since casein is highly abundant in milk sources, through rennet. And then this casein removal is to be followed by tangential flow filtration, which was demonstrated to be a reproducible platform for functionally competent EVs, where functionally competent is to be underlined again for downstream applications. So finally, after covering also these real case scenarios, I would like to summarize what I have been discussing regarding TFF. First of all, TFF or tangential flow filtration provides multiple advantages to overcome the unmet needs currently in the field of extracellular vesicles when it comes to isolation of EVs. And TFF is a candidate to do so basically by the help of its versatility, which allows exploitation in multiple scenarios. And because of this versatility, it is becoming more and more recognized because it can be used in very different configurations in many different application scenarios. And with the field now moving towards more application phase, either industrial or clinical from basic research, we can expect to hear TFF even more in the coming years because its advantages are even more pronounced when it comes to large-scale production or manufacturing of EVs. So if you are interested in any further reading, here are my references that I used throughout my presentation. If you have any questions, I would be glad to answer them, and I would like to also thank you for your attention.